So right, the concept of electronegativity is a concept that means how easily an atom is able to give up its electron, and how tightly it is holding on to its electron. So the atoms typically in the upper right-hand side of this periodic table are the most electronegative, and the ones as you go across and down are less electronegative. And so you can use that concept of electronegativity to figure out, give you an idea about, excuse me, how that atom, what kind of bonds that atom will form with other atoms, whether it will be forming a covalent bond where the electrons are equally shared, or will it be forming an ionic bond where it is completely donating its electron, or completely accepting an electron to form an ion, either a cation or an anion. And you can also look at the difference in electronegativity between two atoms to figure out what kind of a bond they might form. And we also talked about the types of intramolecular bonds. We talked about ionic bonds and covalent bonds. And under covalent bonds, we have the nonpolar type or the polar type. We discussed some of those. We discussed this example where you have the polar covalent bond, where although it is a covalent bond, there is a polarity, so that one of the atoms has got a stronger pull on the electrons. So it will have a partial negative charge at this end of the molecule. And as a result of the hydrogen's electron being more strongly attracted by the chlorine, it will have a partial positive charge. And so this would be considered to be a polar molecule. We talked a little bit about molecular shape. And we discussed a little bit about the geometry of different types of molecules. And in one of your assignments, we discussed the difference in geometry between an ammonia versus an ammonium molecule. And essentially that is the situation here with these two molecules here. Okay, where you have this pair of unbonded electrons that is pushing down on these bonds, so forming that pyramidal type of geometry. And I think most people got that. I think actually maybe all three of you were okay with that question. And then right, we talked about now polarity of the molecule overall. And we discussed that whether the bonds are polar or not polar, anytime you have a symmetrical molecule, because everything is symmetrical and everything is equivalent, they always produce non-polar molecules, whether or not the bonds between their atoms are of a polar nature or a non-polar nature. And whereas in asymmetrical molecules, it just depends on the type of bonds and where those polar or not polar bonds, how they are arranged. overall, an asymmetrical molecule may either be polar or non-polar, depending on the bonds and the geometry. Next, we discussed a little bit about those intermolecular bonds, whereas intramolecular bonds are bonds within a molecule, between the atoms within a molecule. But then intermolecular bonds, these are bonds between molecules that are outside of the molecules and between the molecules. And we discussed three types: the London dispersion forces, the dipole-dipole forces, and the hydrogen bonds. We discussed the different characteristics of all of them, and we discussed how London forces were present in between all molecules. And that in terms of increasing strength, these London dispersion forces were the weakest, whereas the dipole-dipole forces were slightly stronger than the London dispersion forces, but not as strong as the hydrogen bonds. And finally, the hydrogen bonds were the strongest of the intermolecular bonds. Okay, we talked a little bit about the properties of water. We talked about its importance in terms of being a universal solvent, because it's able to dissolve many different types of solutes, and it's universally present in almost all types of life forms, and it's present in the cytoplasm of cells, and it's really the medium that holds all the molecules together in the cell. We also talked about this is due for it having polarity in the molecule. It has a partial positive and partial negative charge, and that is mostly the reason why it is such a good universal solvent. We also talked about the adhesion and cohesion properties of water. And we talked about the very high specific heat capacity of water and how it helps organisms to maintain a constant body temperature, where the temperature outside might be shifting and fluctuating quite a bit. This specific high capacity helps to somehow equilibrate the body temperature of organisms. We talked briefly about the pH scale, how it runs from 0 to 14. And I think most of you should know that any pH below 7 is considered to be acidic, 7 being neutral, and anything above 7 is considered to be basic. And in water, you have the arrangement where you have an oxygen molecule bound to 2 hydrogen molecules, but when in its purest form, it is able to separate into hydrogen ions and hydroxyl ions. And this hydrogen ion without any electrons is referred to as a proton and has a positive charge. The hydroxide ion has got a negative charge. And when the two combine together, they neutralize and form a neutral molecule of water. And what else? We continue to talk about, excuse me, chemistry of life. We talked about functional groups, and I hope you guys are familiar with all of these functional groups by now. Certainly those of you who have already taken grade 12 chemistry should be very familiar with these. You should know these functional groups inside and out. The OH group, that's an alcohol group, also known as a hydroxyl group. The C double bound to O and an H, that's known as a carbonyl group, and it's found in aldehydes. And in ketones, this group occurs in the center of the molecule, so that it's carbon double bonded to an oxygen, and instead of a hydrogen, it is bound to the rest of the molecule, whatever it may be, to more hydrocarbons here. And in this type of an arrangement, it is referred to as a ketone. An acetone, for example, would be one of them. Oftentimes molecules that are ketones end in the ending -one, such as acetone. Molecules that are aldehydes end in the ending -al, such as this example, propanal. And of course, alcohols end in OL, such as ethanol, methanol, propanol, etc. Then we talked about these two important groups, the carboxyl and the amino groups, and these became actually very relevant when we talked later on about proteins, because all proteins are made up of amino acids, and amino acids have got a carboxyl group at one end and an amino group at the other, such as this amino acid, this glycine molecule. This is the smallest one of the amino acids. Amino acids, if you recall, have got the central carbon, the carboxyl group on one side, and the amino group on the other side. The central carbon is bound to one hydrogen, and then the other bond is an R group. An R group can be any number of different types of atoms in the molecule, in the amino acid glycine. This R group is just the hydrogen. So that makes glycine the smallest amino acid out of all the other 20 amino acids. There's also sulfhydryl groups, SH group, and these molecules are known as thiols. Mercaptoethanol is a good example of a thiol molecule. It has this SH group at its end. Phosphates, very important. We talked about phosphates as relevant to DNA. DNA has got phosphates in its backbone. Its backbone is made out of sugar groups and phosphate groups. Phosphate is a part of ATP and ADP, adenosine triphosphate, adenosine diphosphate. And also another example of this is glycerol phosphate. And you can see the phosphate group over here. Right, there was a tip here about remembering the bonding capacity of these common molecules. And if you don't know it by now, this honk might help you remember it. 1, 2, 3, 4 is the bonding capacity of hydrogen, oxygen, nitrogen, and carbon. We covered redox reactions, and we used the acronym LEO, the lion goes grr, to help us remember that anytime we're talking about oxidation, oxidation is a loss of electrons. So at any time when you have something that is losing electrons, it is going through oxidation and becoming oxidized. So if you have this compound A with 2 electrons and it loses these electrons to become A without the 2 electrons, it is going through oxidation, it is becoming oxidized. And typically, oxidation and reduction reactions occur in pairs, so that this loss of these 2 electrons from compound A it is lost to something else that is gaining the electrons. In this case, compound B is gaining these electrons. Here it is without any electrons, and once it gains these electrons, it becomes compound B plus 2 electrons on it. It is going through reduction and becoming reduced. Anytime we're talking about reduction, we're talking about a gaining of electrons. You're gaining electrons. And so remembering Leo the lion goes grr helps us remember loss of electrons, oxidation, gain of electrons, reduction, Leo and grr. And we also saw these types of oxidation reduction reactions be very important in when we were looking at photosynthesis and cellular respiration. We continued looking at biological macromolecules, carbohydrates, fats, proteins, and nucleic acids. We discussed these in terms of the subunits and the monomers that make up these macromolecules, such as monomers of monosaccharides, form larger molecules of disaccharides or polysaccharides or oligosaccharides, which are in the carbohydrate class. Excuse me. Under fats and lipids, we have the smaller molecules of glycerol and fatty acids that then, when they come together, form these larger diglycerides or triglycerides. And these are the macromolecules that fall under the fats or lipids category. And then proteins, of course, amino acids are the monomer subunits. And when you have multiple amino acids joined together through peptide bonds, you get a string of amino acids, which we call a protein, a peptide, or if it's larger and has a function, it's a protein. If it is catalyzing a reaction, we call this an enzyme. We have somebody joining us. Excuse me. Hi, Nagar.
Hi, sorry.
That's all right.
I was working.
Okay. And then finally, we talked about nucleic acids. We didn't just touch on it. We had a whole many lessons devoted to nucleic acids, the DNA and RNA, and we discussed how those are made up of the smaller monomers of nucleotides. We talked about condensation and hydrolysis reactions. We talked about how condensation reaction, or another name for condensation reaction, is a dehydration reaction, and how these were synthesis types of reactions. So for example, you have these two monomers, these two sugar molecules, simple sugars, a glucose and a fructose, and you can form the molecule sucrose through a condensation reaction, also known as a dehydration reaction. Why is it called a condensation or dehydration? Well, what do you do when you're dehydrated? When you're dehydrated, you have lost water. What happens during condensation? When you have condensation, water is falling out from vapor. Water is forming. So that's exactly why this reaction is called a condensation or a dehydration reaction, because a molecule of water gets eliminated when this bond gets formed. So you see there's the hydrogen and then an OH group. Together they form an H2O molecule, and then this bond called the glycosidic linkage is made. And you have this dipeptide called sucrose. And then So this type of reaction is known as a synthesis reaction or an anabolic reaction. It creates bonds and it makes larger molecules from smaller molecules. And then the reverse is true. The reverse is true in a hydrolysis reaction. In a hydrolysis reaction, it's a catabolic reaction. It's a breaking down of a molecule into smaller, a larger molecule into a smaller molecule. So the hydrolysis molecule is the exact reverse of this condensation dehydration type of reaction. And it's a hydrolysis reaction. And if you think about the term hydrolysis, hydrolysis, the hydro part refers to water, and lysis means to break. So you're using a molecule of water to break the bond. So, so this larger molecule plus water breaks this bond, and you get two smaller subunits formed, two smaller molecules formed. So hydrolysis and condensation are reverse reactions of one another. One involves the elimination of water, the other involves the addition of water in order to break a bond. We've already discussed a little bit about carbohydrates and their monomeric units. Oligosaccharides, 2 or 3 simple sugars bound together. Polysaccharides are several hundred to thousands of these monosaccharide units joined together by the glycosidic linkages, that type of linkage that we talked about up here. That's a glycosidic linkage between 2 sugar subunits. And of course, you have these enormous molecules of glycogen, cellulose, and there's examples of them here: glycogen, starch, cellulose. And we talked a little bit about those, how they can be highly branched. We talked about the diglyceride or triglyceride and how they're made, how the individual subunits, what those are made from. So here we have a triglyceride that in this reaction, in this forward direction, that is a hydrolysis reaction. Again, the breaking of a bond by the insertion of water molecules. So a water molecule is added to break these bonds. These are ester bonds here that are broken. And once it's broken, you have the fatty acids and the glycerol part of the molecule. And the reverse of this would be the condensation reaction, where you are removing this water in order to create these bonds and to create the larger triglyceride molecule. And under fats and lipids, we also talked about phospholipids and how phospholipids are very important because those are the units that make up the cell membrane. And we talked about what is special about the phospholipid that makes it so well suited to creating a membrane, a bilayer. And what's special about it and what's important about it is that it has a polar or hydrophilic head group. So this polar hydrophilic head group here, because it's hydrophilic, it is water loving, this polar head tends to want to face outside to the water. So when you place these phospholipids in the water, they automatically, to be energetically favorable, will face their polar hydrophilic parts out towards the water, and then the hydrophobic or the non-polar hydrocarbon tails want to hide from water. So they will kind of find and face each other and avoid being in contact with water. So as such, they form this very stable lipid bilayer. This, when you add phospholipids to water, they form these spheres called micelles, where all the tails are tucked in and all the heads are sticking out. And then we covered proteins with quite a lot of focus on the amino acid groups and the importance of the amino group, the carboxyl group. And this is the basic structure of all amino acid types. There's a central carbon, and carbon has 4 bonds, 1, 2, 3, and 4. On one side, it has the amino group. On the other side, it has the carboxyl group. On the third bond, it has a hydrogen. On the fourth bond, this is where the variation happens. This is where you get different amino acids depending on what you have here on this, what you call an R group, because it can be a number of different things. And that's what makes the 20 different amino acids have different kinds of property depending on what they have at this fourth position on carbon. And then this is all the different types of amino acids that you can get. If you have the very basic, just a hydrogen there at the R group, you get your most simple, most basic, where is it? Glycine. So here at the R group, if you only have another hydrogen, you get a glycine molecule. And this, as you can see, is the smallest amino acid. Suppose instead of a hydrogen, you have a CH3. You have a methyl group, then you will have the amino acid alanine. It is slightly larger than the glycine. So depending on what you have in this position of the R group, you have different amino acids. And these side groups, these side chains, give different properties to these amino acids, such that you can have amino acids where the side chain, the R group, might be hydrophobic, as is the case with these bunch of amino acids. And if you look closely at their R group, you can see they're mostly hydrocarbon. There's no charge on them. So these are hydrophobic. They don't like water. And at the positions where these amino acids are found on a protein, they tend to avoid the water phase and want to be facing more to the interior of the protein. You have the hydrophilic side group. So if you look at the side groups here, you see that there's polarity on them. They're charged, or they have oxygens, they have charges or amino groups, or so these will be polar. These would be hydrophilic. These would be water-loving side chains, and in a protein, they would be quite happy to be facing the watery outside part of the protein. And then you have some amino acids that properties are somewhere in between. They kind of can have both kinds of features, are happy in both kinds of environments, based on the character of their side chain. We talked about the structural hierarchy in proteins, how you have 4 levels of structural complexity. At the most basic level is the primary structure. And the primary structure, excuse me, the primary structure, all it really is the order of the amino acid groups in the polypeptide chain. So what follows what? That would be your primary structure. So for example, if we were to look at this diagram here, in A, depending if we imagine each one of these little balls here is an amino acid, and depending on which amino acid follows what, this would be our primary sequence. For example, we might say we have a valine, we have a glutamate, we have a proline, we have a glycine, and so that order would be our primary structure. And then based on that, the next level of complexity that we have, if you recall, was the secondary structure. So as you guessed it, that's how it goes: primary, secondary, tertiary, and quaternary. And in the secondary structure, because of the ability for these amino acids to form hydrogen bonds with each other, excuse me for a minute while I have a drink. And because, remember, these amino acids in their backbone have got the amino and the carboxyl groups. And so these groups are able to form hydrogen bonds with each other. So in order to, and because of this hydrogen bonding, they're able to form some very stable types of arrangements, which we call secondary structures. Sometimes they're able to form this right-handed coil we call an alpha helix. And our hair protein is almost completely made out of this type of secondary structure, the alpha helix. And depending on the sequence, and sometimes they're able to form beta pleated sheets. And silk is a good example of something that has a lot of this type of secondary structure. It's a large amount of beta pleated sheets in silk, and that is probably why it's so smooth, because it has all these amino acids arranged in very smooth sheet-like structures. So those, if we look at this picture, would be like this. So here's that right-handed coil, the helical coil called the alpha helix. And if we look closely, I'll try and zoom in so that I can point. So you can see this dotted line here shows the hydrogen bond across the coil that's holding the peptide chain in this coil-like arrangement. It's this hydrogen bonding between the carboxyl group from this amino acid up here to another amino acid, a few amino acids down to the amino group of that one. There are these hydrogen bonds, and they stabilize this alpha helix. So this is one of those secondary structures, and the other one is that beta pleated sheet, where you have two strands of the polypeptide, and this would be one, and this would be another. Sometimes it's the same one, and it's just going down and making a turn, almost making like a hairpin loop. It's making a turn down here, and then across from each other, they're able to form these hydrogen bonds that stabilize this type of structure. And it almost forms like a smooth pleated sheet. So it's called a beta pleated sheet, alpha helices and beta pleated sheets, two types of secondary structure. And then these two types of secondary structure, together with various loops and turns, pack together to form the tertiary structure, or the overall three-dimensional shape of the protein or the enzyme. And in some proteins or enzymes, you have more than one polypeptide. So here you have one polypeptide with alpha helices shown in yellow, beta pleated sheets shown in green that needs to come together with a second one that may be the blue and the pink to form the quaternary structure. So the quaternary structure is where you have a protein that has more than one polypeptide coming together to form that three-dimensional overall shape. And hemoglobin is a good example of this type of protein. It is made out of 4 of these polypeptide chains so 4 of them will need to come together in order to form a stable hemoglobin protein. Very, very important concept when it comes to time to proteins is how, when we talk about proteins, their structure, their three-dimensional shape and structure is very important to their function. So the way they work, the reactions that they catalyze, or or the molecules they may help to mediate the movement of, has got a lot to do with their three-dimensional shape. Disrupt that shape, mess with that shape, and you have messed with the function. So in other words, if you have some sort of a mutation where one of these amino acids here might get swapped out for something else, maybe that amino acid was very important in maintaining this beta pleated sheet-like structure. And if you put a different amino acid there, then you won't have that beta pleated sheet. You might get a kink in there, you might get a loop in there, and that beta sheet no longer fits into the overall three-dimensional shape of that protein, of that enzyme. And by not fitting in there anymore, it is now not able to adopt the right three-dimensional shape to catalyze that reaction effectively. Those side chains that need to carry out that chemical reaction are no longer positioned in the right orientation and are not able to form the right bonds in order to form and break bonds and catalyze the reaction. So if you disrupt your protein's structure, you have disrupted oftentimes your protein's function. So in proteins and enzymes, structure and function are very tightly related. And a good example of this we talked about was in sickle cell anemia. So one way of disrupting the structure was, for example, through a mutation. That might change the sequence of the protein. But other than mutations, obviously these proteins can also be denatured if they are in outside of the pH range or temperature range that maintains that proper shape, that is energetically favorable to maintain that shape. If a protein loses that shape, that normal shape, it is said to be denatured and it will no longer function properly. One example of this is if you look at egg whites. We all know what egg whites look like when they're not denatured. That is when they're raw and they're quite clear and gelatinous. But go ahead and heat them by boiling them or frying them. You have denatured them and you've turned them very hard and solid and white. That is an example of a protein that is being denatured. I guess same with any type of a meat that you might cook. That protein, once it gets cooked or denatured, is very different than when it was in its natural state and before it was denatured and cooked. So we've already talked about hydrolysis and condensation reactions, but it's also important to relate those reactions to the formation of proteins, where you have two single amino acids forming a bond together, this peptide bond. And a water molecule is lost. Anytime you have a reaction that a water molecule is lost in this way, this is called a condensation reaction. And through the loss of that water molecule, a bond is formed and a bigger molecule is also formed. From two small amino acids, you get a larger dipeptide. And you can have a third amino acid join in the same way. Loss of a molecule of water, a peptide bond formed, and now you have a tripeptide, and so on and so forth. And before you know it, that's how you have a growing chain of amino acids, a growing polypeptide. And at one end of that chain, you will have your amino group, your free amino group, and at the other end of the chain, you have your carboxyl group. Then our discussion carried on more detail about nucleic acid. And we looked at the two types of nucleic acid, deoxyribonucleic acid and ribonucleic acid. We talked about how the structure of a nucleotide, there is a sugar, a ribose sugar, there is a phosphate group, and then there is a nitrogenous base, and that's what makes the nucleotide structure. And depending on the type of nucleotide base, you would have different types of nucleotides. Then we paused there with our discussion of nucleic acids. We continued this further when we talked about our genetics unit. We paused here and we went into a little bit of cellular biology just to familiarize ourselves with all the different parts of a cell. We learned about the different types of cell, the eukaryotes versus the prokaryotes. We learned a little bit about the basic structure, cell structure of a prokaryotic cell, and the basic structure of a eukaryotic cell. And we learned about some of the important cell structures within a eukaryotic cell, and especially as we learned more about the copying of DNA, the transcription and translation of DNA. We learned about these various loci in the cell, the nucleus, the ribosome, and the endoplasmic reticulum. We learned a little bit about the function of these different organelles and how they are related to the overall functioning of the cell. We looked at a couple of examples of eukaryotic cells. We looked at the animal cell versus a plant cell and noted that they have very much similar structures, similar organelles with certain differences. We continued to look at the structure of the membrane and the phospholipid bilayer. Again, important to note that there's the polar head group and there's the hydrophobic hydrocarbons that are non-polar, and they tuck in order to avoid that watery inside and outside of the cell. If they face each other, they can exclude water and they don't have to come into contact with water. We talked about the model of the phospholipid bilayer, the fluid mosaic model. we talked about the movement of molecules across the plasma membrane. And also, I went ahead and added in a few more YouTube links for you guys to watch related to these lessons about the phospholipid bilayer, the movement of molecules across the phospholipid bilayer. There's different YouTube links here that you guys can watch just to jog your memory and perhaps renew your interest when you're studying these concepts and reviewing them. We talked about movement through diffusion of molecules from an area where they have a higher concentration to an area of lesser concentration. We talked about how this diffusion from high concentration to low concentration is a spontaneous process. It doesn't require any input of energy. The only thing it does require is for there to be a difference in the concentration, so a concentration gradient. And water being one of those molecules that is able to diffuse quite freely across the plasma membrane, and the special name given to the diffusion of water across a concentration gradient from one side to the other side of a plasma membrane is called osmosis. We talked a little bit about different types of solution depending on how much solute was in the solution, whether or not it would be an isotonic, hypotonic, or hypotonic type of solution, and the effect that it might have if a normal cell was placed in this type of a solution. In an isotonic solution, it would be quite happy. In a hypotonic solution, it would lyse as a result of water wanting to rush in from an area where there's higher concentration of water to an area where there's lower concentration of water. The cell would plump up and eventually burst. And in a hypertonic solution where there is less water out here, water would want to leave the cell in the direction of the concentration gradient, and this would lead to the shriveling up of the cell or crenation. So diffusion happens quite freely. And water is one of those molecules that can freely diffuse, and it doesn't need help from anything else to go across the membrane. All it needs is a concentration gradient. But then you also have protein molecules that need carrier proteins to help them diffuse across the membrane to get into the one side or the other of the cell. And these carrier proteins can be very specific to certain types of molecules that they need to transport. We talked about the two types of transport that you can have. You could have a passive type of transport, or you could have an active transport. So during this type of passive transport, no energy input is required. All you need, again, is just a concentration difference between the two sides. So in this case, a molecule like water doesn't need anything to facilitate its diffusion. It can just freely diffuse across the membrane. But you might have different types of molecules which need the help of these transporters to help to diffuse, to help them pass across the membrane from the area of higher concentration to an area of lower concentration. But during active transport, you need to use, to spend energy and to use these transporter molecules to transport molecules in the opposite direction to the concentration gradient. So you're putting in energy to push these molecules or ions from a side where there is already not much of them to a side where there is already a high concentration of them. And you're continuing to pump these across to build up even more concentration on one side. And in order to do that, you need to use active transport and use energy from an ATP type of molecule. And the sodium-potassium pump is a good example of this type of pump. And it's very important, as we'll learn later on in one of our future units, for transmission of nerve impulses across nerve cells. Sodium-potassium pump. There's a couple of YouTube videos that you guys can spend some time and watch. It might help. We covered endocytosis and exocytosis. And finally, in our lesson 4, our focus was enzymes. We talked about exergonic and endergonic reactions. And we looked at these definitions of what does exergonic and endergonic mean, and whether exothermic and endothermic, what do they mean? So certainly familiarize yourself with these types of energy diagrams. We talked about the enzymatic pathway. We talked about how on an enzyme, there is an active site where the substrate binds, and oftentimes the process of substrate and enzyme binding is happens through a process of induced fit, so that at the time before binding, the active site can somewhat allow for the binding of the substrate, but through the substrate binding in, it becomes an even more perfect fit. It induces a better fit. And so together they form this enzyme substrate complex, which then catalyzes the reaction. In this case, the reaction that is being catalyzed is the hydrolysis of a sucrose molecule. So again, hydrolysis means breaking up of a bond through the addition of water. So plus water, break the bond, and your disaccharide, which was a sucrose, is now 2 monosaccharides, a glucose and a fructose, and your enzyme is available to catalyze another round of a reaction. Again, some YouTube videos for you guys to watch. We talked about the important factors that affect enzymatic activity, how temperature, pH, and the effect of the concentration of both the enzyme and the substrate. So these 4 factors can really influence the rate of and the activity of an enzyme. We talked about the inhibition of enzymes, how enzymes have the active site, and they also have an allosteric site. And the allosteric site is a location on the enzyme, on the surface of the protein, different or other than this active site where this is the position of substrate binding. And so this allosteric site, you can have something binding to it to either make this enzyme active or inactivate this enzyme altogether. We talked about inhibition and the 2 types of inhibition, competitive inhibition and non-competitive inhibition. And these diagrams show very clearly what we mean by competitive inhibition is where you have the competitive inhibitor actually binding to or part of the site that the substrate would normally binding in, and by binding that, it doesn't allow the substrate to bind, so it's kind of competing for this site with the substrate. So once this is there, it doesn't allow this to bind, so it doesn't allow the reaction to be catalyzed. It's a competition as to who gets in there. And non-competitive binding, a non-competitive inhibitor isn't interested in binding to, it doesn't have the right shape or the right features to bind to anywhere on the substrate binding site. But what it does bind to, it binds to a site other than, so to an allosteric site. And by doing so, this binding may induce a change or an alteration in the overall shape of the enzyme or the active site that then no longer allows the substrate to bind in and for the reaction to be catalyzed. So this is non-competitive inhibition. And then we, again, talked about allosteric regulation and how you can have activators or you can have inhibitors. So you can have activators that bind to that allosteric site of an enzyme that will promote the action of the enzyme. And or you could have inhibitors that would bind to the allosteric site and actually suppress and inhibit the activity of that enzyme. We looked at feedback inhibition, where you can have you could have a product, the end product of an enzymatic reaction pathway can bind to the allosteric site of one of the primary enzymes, one of the enzymes further upstream, and actually influence or shut down the pathway. So if you have too much of this, it will bind and that will shut down the pathway until you don't have enough, and then this will come off and the reaction can continue. So this is a form of a feedback inhibition. Then we talked about how you can have enzymes that can have a different active form compared to an inactive form, where they're in their active form, their active site is available, whereas in their inactive form, their active site is not available for binding substrates. And so when you might have a molecule that may act as an activator or as an inhibitor, those molecules may carry out their action by stabilizing, by binding to an allosteric site and stabilizing that active form. So keeping it in that active shape so that the active site is available for substrate binding. Or you could have an inhibitor that may bind to the inactive form and stabilize the inactive form. And by stabilizing the inactive form, it doesn't allow for the active site to be available for binding substrate and carrying out that catalytic reaction. We talked a little bit about all different kinds of uses of enzyme in the food industry, and we watched these videos together in class about what is a protein and how do enzymes work. And that pretty much is our Unit 1. And please, if you have any specific questions, talk to me about your specific questions. Otherwise, I'm just doing my best to give you an overall, go over everything from Unit 1, 2, and 3. So we're done Unit 1. We still have a little less than an hour to go. We can talk about these metabolic processes now. We can talk about thermodynamics. To review thermodynamics, and the concept of bond energy, how you can have reactions that are exothermic, that release energy, reactions that are endothermic, that that consume energy and take up energy. We talked about the first law of thermodynamics, law of conservation of energy, that says the total amount of energy in the universe is constant and energy can't be created or destroyed, just transformed from one form to another. For example, in photosynthesis, you have a transformation of radiant energy, sunlight, transformed into chemical energy to make glucose and sugar and ATP. We talked about the second law of thermodynamics and entropy. So according to the second law of thermodynamics, the disorder in the universe is always increasing with any change that occurs. And so there is an unstoppable trend towards randomness. That was a second law of thermodynamics, thermodynamics. We talked a bit about Gibbs free energy and looked at the equation and how in that equation we have both an enthalpy term, but also a temperature and an entropy term. We looked at the ATP molecule, adenosine triphosphate, so 3 phosphate groups. And here is the nitrogenous base adenine. And here's the ribose sugar, the 5-member sugar ring here. 5 carbon sugar, 1, 2, 3, 4, 5 carbons on the ribose. And how you can have ATP, and you can break the bond between the second and the third phosphate here to free up an inorganic phosphate and some free energy. So you can get ADP, a free phosphate, and some energy, and that's how ATP is an energy molecule. And ATP and ADP go through this cycle where you get the input of energy from exothermic reactions, from cellular respiration, for example, and you create ATP, you make more ATP, but then you use this energy by removing energy for other endothermic reactions. For example, you will use this energy for protein synthesis, for nerve conduction or muscle contractions. And by doing so, you will make your ADP again and the inorganic phosphate, which will then require an input of energy to make ATP, and the cycle goes around and around. Next, we looked at photosynthesis and cellular respiration. And quite basically, we looked at photosynthesis occurring in 2 stages, the light dependent and the light independent stage. We discussed how light excites some of the photocenters in the thylakoids inside the chloroplast and excites an electron and promotes it to acceptors, which will pass it down to further and further down into other acceptors, and as the electron moves through the electron transport chain complex, you get a buildup of hydrogens that are being transported from the stroma into the lumen of the from the stroma into the lumen of the thylakoid. And as a result, this buildup of, whoops, let me do that. These hydrogens that are brought in to the thylakoid lumen, now you have a concentration gradient for these hydrogen ions, or a proton gradient. And the energy from this proton gradient can be used by an enzyme called ATPase, or ATP synthase. And it uses this energy, this proton gradient, as protons move across this protein back to outside to the stroma. That energy is used to create ATP from ADP. And you also have, so you have some ATP being made during photosynthesis, but then you have the light independent stage, or the Calvin cycle, where you go ahead and you use carbon dioxide, and you, in the Calvin cycle, you have 3 steps: the fixation of carbon, the reduction of 3-phosphoglycerate to glyceraldehyde 3-phosphate, which is then regenerated to form RUBP and goes through the cycle again and again. And then you have this glyceraldehyde 3-phosphate, which then gets converted into glucose. And glucose then is a chemical form of energy that can be used. So the energy that was taken up from the sun at the photocenter is now used to create glucose, as well as some ATP. And then in cellular respiration, this glucose can be used to provide even more energy in the form of ATP. And this happens through the reactions under the process of glycolysis, pyruvate oxidation, and Krebs cycle. And finally, all these electron carriers, NADH, FADH2, they all pass down the electron transport chain, and finally create quite a large amount of ATP through this process of oxidative phosphorylation. So all of the details of glycolysis are laid out here. I'm not going to go through them, but I'm not expecting you to have these memorized. I just need you to have an awareness and to be able to be familiar and comfortable with working your way through this and understanding what's happening. And but this isn't something I'm expecting you to memorize. I don't think even at the university level, you might be expected to memorize this. I can't remember. I probably did have to memorize this at some point in university, but I don't think you're expected to retain that unless you are a student who's specifically focusing on photosynthesis or cellular respiration. Then it's important to know that the pyruvate molecules that are made at the end of glycolysis are then and fed into the Krebs cycle once they are converted to acetyl-CoA and attached to this enzyme CoA. And this acetyl-CoA then goes through what is the Krebs cycle. Krebs cycle is also known as the citric acid cycle, and that's because you see citrate here and isocitrate, and it just goes through that loop. If you recall on one of the questions, I was asking in your assignment about what happens if you don't have any of the sugars and instead you have alpha ketoglutarate, what happens there? And basically what I wanted you to understand is that to look for, okay, alpha ketoglutarate, where do we find alpha ketoglutarate? We find it during Krebs cycle. Where do we find it in Krebs cycle? We find it at this point. So if we are feeding alpha ketoglutarate into the Krebs cycle at this point, what happens is that we are producing some NADH. We're not producing this one because this is a step that happens before alpha ketoglutarate. We're producing some NADH, we're producing some ATP, and we're producing FADH2, and again, NADH here. So what happens there is that, can this cycle continue? No, because this cycle needs more of this acetyl-CoA to come in, and this is coming from glycolysis. If there's no sugar, this is not coming in this way. So this essentially by inputting alpha ketoglutarate, you're just doing this process from here to here, stop. So I wanted you to notice that you make one ATP, you get 2 molecules of NADH, then you make 1 molecule of FADH2. And then I wanted you to take those molecules and see for every NADH and FADH2, how many ATP molecules can you make? And that's how you would approach solving that problem. We talked a little bit about the electron transport chain and how the electrons were passed down by these energy carrier molecules of NADH and FADH2, and how the whole complex is located on the inner mitochondrial membrane. And finally, all of these protons are used to, that are built up in the intermembrane space, are transported across ATP synthase, and the energy for this proton gradient, again, is used to make ATP through oxidative phosphorylation. And so by the end of the process, from 1 molecule of glucose, you get a grand total of 36 molecules of ATP, and that breakdown, that energy is breakdown is summarized for you here, a grand total of 36 ATP. We talked a little bit about alcohol fermentation and lactic acid fermentation, and how in the absence in the absence of oxygen, when there's not enough oxygen, these are processes that are available to create at least some amount of energy. And then we basically put the processes of photosynthesis and cellular respiration side by side and understood how they are actually quite interdependent. In plants, you have photosynthesis and cellular respiration happening. And obviously, we are not photoautotrophs. We are not able to carry out our photosynthesis. So we have to get our glucose through consuming plants that have glucose. And so use that to feed our cellular respiration process and to feed our mitochondria. To make ATP for our various processes, we can use our ATP energy to build molecules and proteins and break down fats and build up fats and transport molecules across the cell for all sorts of things. We talked a little bit about nutrition and diets, different types of diets. And we talked about bioremediation and the use of living organisms to help to clean up contaminated soil or water. We also talked about phytoremediation, which was a type of bioremediation that instead of using bacteria, used plants to carry out the cleaning of contaminated soil or water. And that brings us to the end of Unit 2. And finally, Unit 3 was our molecular genetics unit. We introduced you to the concept of heredity and how Gregor Mendel was the first one to actually lay out some controlled experiments where he came to the understanding of the traits of the offspring came from both parents. And we went through and explored how the works of various scientists like Friedrich Miescher, who discovered nuclein, which later became known as DNA, and Phoebus Levine, who discovered the two types of sugar, the ribose and the deoxyribose. And Hammerling, who did the experiment that strongly suggested that the hereditary material was located in the nucleus. The nucleus was what held hereditary material. We looked at the works of Griffith and how he discovered transformation, how you could have a mixture of heat-killed pathogenic strain mixed together with a non-pathogenic strain, and you could get transformation still. Where the heat-killed pathogenic bacteria would mix together with the non-pathogenic bacteria and still be able to kill these mice. And then the group of Avery, McLeod, and McCarty, they were the ones that demonstrated that the transforming substance in that previous experiment was DNA, because up until that point, it was still a question as to whether it was the DNA or protein that was the transforming principle. We talked about Chargaff's rule and how Erwin Chargaff discovered that you had always equal amounts of A's and T in the DNA molecule of any particular organism. And that really laid the groundwork of the base pairing rule in the model of DNA, where DNA A always pairs up with T and G always pairs up with C. And then there was the Hershey and Chase experiment, which showed quite definitively that DNA is the hereditary material. They did that experiment by using they used a virus particle, and they labeled with radioactive phosphorus, which would tag DNA, or they're labeled radioactive sulfur, which would tag the protein coat of the virus. And so by doing this experiment, they were able to show that the radioactive phosphorus was found within the cell and the radioactive sulfur was found outside of the cell. And this firmly established that that genetically inherited material that entered the cell was the DNA. Then the model of the DNA was built by Watson and Crick, which used Rosalind Franklin's very important X-ray crystallographic experiments, the X-ray diffraction patterns of DNA that she collected and analyzed, and was able to determine that there was a certain rhythm and repeatability to this structure. And from that, they were able to together build the model, the double helix structure of DNA, where the backbone is made out of sugars and phosphates, and there's a directionality to the strands. The two strands run anti-parallel to each other, whereas one runs from 5' to 3', and the other one would run from 5' to 3' in the opposite direction. And in the middle, if you can imagine, if you think of this as looking very much like a twisted ladder, so the rungs of the ladder would be the nucleotide bases that would then hydrogen bond quite stably across the two strands with one another. And to note here, A's and T's always bind with each other, G and C always bind together. And guanine and cytosine always have got 3 hydrogen bonds, making the bonds between C's and G's much stronger than between A's and T's, which only have 2 hydrogen bonds. There's again YouTube video you can watch. DNA replication and repair. We talked a little bit about the idea of, so what type of DNA replication happens? Is it conservative, semi-conservative, or dispersive? And it was the experiment of Meselson and Stock that showed that replication takes place semi-conservatively. They went ahead and labeled DNA and went through rounds of replication after first generation and second generation, and they spun these DNA in the centrifuge to see light and heavy strands depending on how they were labeled, and they were able to show that in fact this was happening in a semi-conservative manner, where each strand would be used as the template to create the next set of strands. And just pictorially, if you were to show it, if you have a strand of DNA, this is not the scenario that takes place. Rather, this is the scenario that takes place. It happens semi-conservatively, so that each half of the DNA strand is used as a template for making the new other half of the strand. And this happens because you have that complementarity between adenine and thymine and guanine and cytosine. You can review the replication process and the role of each one of these proteins and enzymes and what is their job. What is the job of DNA helicase? What role do the SSBs carry out? What does DNA gyrase do? DNA polymerase, you certainly should look and kind of have an idea and understand the whole process of DNA replication. and the different role of these enzymes. You should understand that there is the leading strand, and then there is the lagging strand. And you should understand that the two strands of the DNA run anti-parallel to each other in opposite directions. And you should know that there are these Okazaki fragments. What are Okazaki fragments? What is the job of DNA ligase? How is DNA polymerase 1 different from DNA polymerase 3? This is DNA polymerase 3. There's DNA polymerase 1. How are they different? And then DNA repair, this proofreading mechanism. Again, there's some YouTube videos for you to watch to help hopefully stimulate your memory. There was this DNA extraction activity, which would have been interesting if any of you did it, or perhaps you can just go through it as a thought experiment and think about it. Then we carried on in the next lesson to look at, okay, so we looked at DNA replication. What else happens with DNA is protein synthesis. That is ultimately another, the most important role of DNA is that it encodes information that has instructions for creating proteins, and the proteins are the actual machinery that carries out the function in the cells. We talked a little bit about the one gene, one polypeptide hypothesis, where you have one gene encoding one polypeptide, which carries out a certain function. And we talked about the central dogma of molecular genetics, where it represents the entire process from DNA to transcription in the form of mRNA. The mRNA leaves the nuclear envelope where it gets translated into a polypeptide, and the polypeptide folds to a protein having the task, be it structural, be it catalytic, be it an enzyme. And that is the central dogma of protein synthesis. We looked at the genetic code. We analyzed codons. We looked at transcription, how during transcription the mRNA is created, and it's done so under stages of initiation, elongation, and termination, followed by finally a fourth stage of post-transcriptional modification. So essentially in initiation, you have your RNA polymerase that binds to that double helical DNA. It binds a promoter region. This promoter region is typically an area that is very rich in A's and T type bases. And the A's and T's, if you recall back that diagram where I was pointing out that those two are only held together with 2 hydrogen bonds. So strand is unwound, the double helix is disrupted, and the template strand is exposed. Then you get some elongation. You get mRNA being made using the template strand, and it's synthesized in the direction of 5' to 3'. But in this case, in mRNA, you have to remember that you will never see any thymines. In this case, the base uracil replaces thymine, and U is complementary to A. Uracil is complementary to adenine. What else? So transcription happens as this mRNA is being made until finally at termination, this RNA polymerase reaches the end, the termination sequence at the end of the gene. And the mRNA and the RNA polymerase are released from the DNA double strand closes again, and you have the polymerase and the mRNA polymerase is free to go and bind to the promoter again and carry out again another set of transcription, and the mRNA then goes into transcriptional modification. And then if you remember, transcriptional modification is that in eukaryotic cells, you have sequences called exons, which are non-coding pieces of DNA. And before this mRNA is able to be translated into protein, these non-coding pieces need to be excised out. You also need a 5' cap and a poly A tail added to stabilize this mRNA. And finally, when all three of those steps are done, you have an mRNA transcript that is ready to be transcribed. It's ready to leave the nucleus and to be transcribed. Now to be translated, not transcribed. Transcription happens when you go from DNA to mRNA, and from mRNA to protein is the translation. It gets translated. So now in translation, this is the part that takes place outside of the nucleus, takes place in the cytoplasm. And in order for this to happen, the mRNA message needs to be read, and then the right amino acids corresponding to each codon need to be put into place and strung together. And by the way, this is now unit 3, and our exam is next Wednesday, so a week today. If you guys hand in your assignments to me on time or even early, then I will have enough time to go ahead and mark those assignments so that you know if you did your assignment correctly or not. So please make sure you hand it in at the latest by Sunday, October 30th, and then hopefully I can mark them and have them to you in a way that you can actually find it useful to go over those questions and answers, and to see if you know everything that you think you know, and if you have all the right ideas when it comes to these concepts. So we talked about the different types of RNA molecule. The tRNA molecule, this is the transfer RNA molecule. It has this type of a cloverleaf structure, and the bottom arm of the tRNA, that's where that anticodon triplet is located. So that if on this anticodon triplet. This corresponds to, it's complementary to whatever is the codon on the mRNA. And so this would have the right amino acid that it will be bound to, and this will bring in the corresponding amino acid by the anticodon binding and corresponding to the codon on the mRNA. So this translation takes place using tRNA molecules, but it also takes place on the ribosome. And the ribosome is composed of 2 subunits. Just trying to think, why is things shifted here? It has 2 subunits. It has the small and the large subunits, and they are made up of various proteins and also the rRNA molecule. And they have these binding sites on them. And there's the P site, or the peptide binding site, and then there's also the A site, or the acceptor site. So how does the process start? Again, as you might suspect, it starts with initiation. Next step is elongation. So you get the next amino acid coming into the acceptor site. These anticodons here recognize the codon sequence on the mRNA and come and bind it, and they have the proper amino acid that corresponds to that codon attached here. And once that coding recognition happens, then through the energy of GTP, 2 GTP molecules, the entire chain of amino acids from the P site, from the tRNA that was located on the P site, get transferred to the next amino acid through the formation of a peptide bond. And so now the entire chain is bound to this tRNA, which is sitting here on the A site. And through, again, the energy of GTP, you get a translation where the entire ribosome shifts along to read the next set. And so now the tRNA that was carrying the rest of the chain that was sitting in the acceptor site is now sitting in the peptide site. So this acceptor site is now ready for the next tRNA to come in, recognize, bind the entire peptide sequence, gets a peptide bond formed with the next amino acid, and it gets shifted here. And again, the cycle goes around and around until this chain of amino acids is made according to the sequence of codons that is read here on this mRNA molecule. And finally, for termination, since there are no tRNAs that code for the stop codon, once the stop codon is arrived at, there is a release factor that binds to the acceptor site. And then when this peptide tries to form a peptide bond, here there is no amino acid here, and so it simply causes the chain to come off of this tRNA and simply float away. And once this floats off, once this amino acid chain moves off, it just signals the disassembly of the whole ribosomal complex, and the ribosome is able to go and bind to the next mRNA and to initiate translation all over again. And there's a YouTube video that you can watch. We talked about different points of regulation of protein synthesis, and that this can happen at 5 control points. This can happen pre-transcriptionally. This can happen at the transcriptional level, post-transcriptional, translational, and post-translational level. We talked about the lac operon. The lac operon is important because it just demonstrates the concept of gene control and how in prokaryotes you have these genes that are organized in a cluster called an operon. So this kind of an operon, lac operon being a good example of this, is one way where they can actually just transcribe and translate everything together, and they're all under the same control of the same promoter, of a single promoter and an operator region here. So there is a YouTube video again here that will help you jog your memory on the lac operon. And we talked about mutations, the different types of mutations, how you can have point mutations that can result in either silent mutations or can cause missense mutations, or nonsense mutations where there might be a stop codon introduced, or there might be frame shift mutations that may happen as a result of an insertion or a deletion of a nucleotide and causes the entire reading frame to get shifted. So that means that all the codons beyond a certain point of that mutation now are affected and completely changed. And here in this figure, and this is directly from your textbook, it shows what types of mutations could possibly happen, just explores different scenarios and how different things can, different types of insertions or deletions or point mutations, what kind of an effect it can have ultimately on the final protein product. Then we talked about biotechnology and gene modification. We explored some of the tools that are available. We explored the use of restriction enzymes. We talked about how restriction enzymes are very specific to recognizing a certain sequence. And what they do is they cut that sequence. Sometimes they cut it in a way where you have these sticky ends. Sometimes they cut it in a blunt way. So depending on the restriction enzyme and the cut site that they recognize. And how these recognition sites are palindromic. So if you look at it, reading it from 5' to 3', it's GAATTC. And on the opposite strand, it's exactly that, GAATTC. And it's like that for all restriction enzymes. It's like that. And it shows where it would cut. For example, EcoR1 would cut between the G and the A, and the G and the A. So once it's cut, you get this sort of a strand. And these ends would be looking to bind to complementary strands, having that same code, that same sequence there. Whereas, for example, small 1 would cut a blunt end. It would not have any sticky ends. We talked about gel electrophoresis, and we looked at how it took advantage of the negative charge that is carried by the phosphate groups of the DNA molecule and how it uses the fact that it can use this negative charge and the size of the DNA fragments to separate them along the gel using an electric current. So the smaller DNA fragments move faster than the larger ones as they move towards the positive electrode, because DNA has got an overall negative charge, right? Yeah, YouTube video again to refresh your mind on gel electrophoresis. We talked about plasmids and how they can be used. They are part of the toolbox for molecular biology, and how plasmids, together with restriction enzymes, can be used to clone a gene into a plasmid and then to use it to get it into a bacteria and get the bacteria to start producing, multiplying and copying and producing that protein. We talked about restriction fragment length polymorphisms, and how they were important in being able to identify a DNA fragment pattern, for example, in DNA fingerprinting, and how this is important in non-coding regions of DNA, because where you have coding regions of DNA, as you expect, it would be nearly identical amongst all of us, because all of us need to produce the same hemoglobin. There cannot be too much changes, otherwise then you get a disease, for example, such as sickle cell anemia. But in our non-coding parts of our DNA, it's not so well conserved, and you can have some unique nucleotide sequences in that area. And if you can imagine, if you digest your DNA with enough different restriction enzymes, then especially in those areas of non-coding DNA, you get enough variation that you can almost form a unique fingerprint of specific individuals. And the whole RFLP process is just diagrammatically summarized here for you, and here in this table explained. We went through the PCR, the polymerase chain reaction, and looked at the process of how you can go from having a small amount of DNA and amplifying it to millions and billions times more. We looked at the DNA sequencing method, both the old-fashioned way and the new modern way. And we ended things by thinking about the social, ethical, and legal implications of biotechnology. And we watched a very thought-provoking, I hope, video together. And we discussed GMOs and gene modification, the pros and cons, the advantages and disadvantages. We talked about gene therapy. We talked about cloning. And I don't have it in these notes, but I certainly, for future, next term when I teach this, I will certainly have a section where I will cover CRISPR technology, because it's been around for a couple of years now at least, and I think it's good to be familiar with it. You don't have to know it, just be aware of its presence and utility. And that brings us to the end of our review session. We are allowed to make a cheat sheet for this exam. Sure, yeah. The answer is yes, you can bring a cheat sheet. It has to be handwritten. A handwritten cheat sheet. So either pen or pencil, nothing that's printed out. Just a handwritten regular 8.5 by 11 sheet. You can fill both sides if you like with whatever you need. And yeah, you can bring a cheat sheet. No problem. I'll send out an email to all the students in the class so that everybody knows that, sure, yeah, I can allow you a cheat sheet. Any other questions, guys? Okay, well, then I think we can stop recording.